Well, for the benefit of the video recording that was not possible yesterday, let me just summarize a bit what we did yesterday, and probably this is also for the benefit of the audience. So what we wanted to do is actually to present several models that have to do with this problem of migration and invasion of tumor cells in regions that are filled of extracellular matrix and fibers, especially when they get too thick. And the typical examples that I mentioned is the one in which the tumors or any tissue is contained in what are called basal membranes. Okay? And these are some examples of them. And I mentioned that the crucial point is the fact that at some point this membrane ruptures, and so what is inside goes outside, and this is the invasion mechanism. Or part of the cell will go outside and colonize the environment. Okay? So the first thing to look at is the behavior of a single cell in this dense environment. And from the experiment you can see that the biggest role is played by the nucleus, which is the stiffest organelle of the cell. So in order to mimic, say, to understand how the cell can crawl and deform the nucleus in order to penetrate in narrow channels or in what they are called constriction rings, you have to understand some mechanics of that. And in particular, you can relate, say, the energy that is required to squeeze this nucleus, and this energy has to be compared with the work that the cell is able to do by traction forces, active traction forces. Okay, you do that, and at the end of the story, what you find is a criterion that tells you when the cell can penetrate the channel and cannot penetrate the channel, just by inequality in some sense. Okay, in which you have, say, mechanical characteristics on one side, and on the other side you have geometric characteristics. So there is a region in which the cell cannot penetrate, of course, when the radius of the pore is too small with respect to the radius of the nucleus, or vice versa if the nucleus is, for instance, too stiff. Good, but this doesn't tell you anything about, say, the velocity of the cell in this microchannel. In order to do that, you can resort to, say, individual based models, and in particular to cellular postmodel, for instance. And then you can run the simulation and see that you have this typical biphasic behavior, a bimodal behavior, in which you have a maximum bell-shaped, say, behavior of the velocity with respect to the pore size, but also with respect to other quantities like adhesiveness or other things. And so there is an optimal velocity. There is an optimal dimension of the pores in order to crawl. Which is, by the way, the more or less the dimension of the nucleus. Well, and then the last step that I slightly covered and I'm going to repeat today is that at some point we want to use this information in a macroscopic framework, in the sense that we want to model, say, ensemble of cells, multi-cell spheroids with billions of cells. And so it's not, of course, convenient to use an individual based model, but one needs to use some continuous models, some PDE models. Okay. So in order to do that, I introduce a concept which is the volume ratio, which is often used instead of the say, the density, just because you're not weighting the cell, but you are looking the cell at the microscope and you are measuring what is the, say, the percentage of space occupied by the cell. So I'm also taking the occasion to make some parallelism with what was done yesterday, in the sense that was mentioned. I'm going by heart, but if I remember at a certain point, a model like that was written. I don't remember this one, and then there was an S. I don't know, N. And then there was, for instance, this one. This is what is important for me. And then V is equal to the gradient of, no, not phi, but probably there was a P. You can also put here some transfer term, Q between the phases. Doesn't make any difference. So what we saw yesterday twice, actually, in the two talks also by Thierry Collen in the evening, is that this is just a special case of what I mentioned generally yesterday. And I'm going to show you why. Okay? So the idea is that you have this thing that is a volume ratio, and then you write mass balance equation for that quantity. Phi C, here P and S are just the same, they are volume ratio, and you can write down this mass balance equation for all the cells, all the constituents, say, of the cellular component, for instance. Okay, like this. You have two components, and then you write two mass balance equation. The difference between this equation here and this one is that. And that I have a velocity for each component, for each constituent, and here you have a common velocity. So let's do something from the beginning. So this is new, in the sense that let's start with just one single cell. Okay, so I'm looking at just one population, a tumor cell, and I'm also treating that as a population that has a constant density. See, it occupies the entire volume. It occupies 70% of the volume, but this is fixed. So what before was phi C is constant, phi bar, and so this means that the equation simplifies a lot, because of course the time derivative of a number is 0, and you can put that number in the denominator, and then you are left with an equation like this. Divergence of V equal to something, which takes care of the growth and death of the cell. Okay, so this is one equation, scalar equation, one single equation for a vector field. Okay, so you cannot solve that. You need to close the system in some way if you want to use this very simple model. And the easiest way to do that is to relate a vector to a scalar, which means something like this. Okay, something like this. So you are just assuming that your field, velocity field, is a potential. It's a gradient of a potential. This is not such a dumb assumption, because from, say, for instance, fluid dynamics, you know that a velocity field can be split into a gradient plus a curl of something, and you are assuming that this part here vanishes, and the curl has to do with something that rotates. So in some sense, you are saying that the cell rearrange the move in the easiest way possible, in the, say, to reach one place to another. They don't do vortex or anything like that. Okay? It's a plausible assumption that, by the way, I will justify in a couple of slides. Okay? From the mechanical point of view. But take it now for, take this equation now as an assumption. Okay? So if you do that, of course, you end up with replacing the velocity in the first equation, and you have an Helmholtz equation and a problem that writes like a free boundary problem. Why? Because you are looking at an ensemble of cells that are growing. If you want to think of tumors, think of tumors. If you want to think of, say, cells that you are culturing on a plate, or say bacteria or whatever, it's just the same. And you have a border of this tumor. So your equation lives in that yellow part there, which is changing in time. Okay, so you need to provide boundary condition at the interface between, say, the tumor and the rest, the environment, and also how this interface moves. So the way the interface moves is very natural. It's moving because the cells are moving. So the velocity of the boundary has to be equal to the velocity of the cells. Okay, and this gives you Or did you do it really? Okay. Okay, never mind. I'll use the old stuff. Okay, and this gives you that. This is the equation for the interface. Okay, because this tells you that the interface moves with the velocity, because the gradient of psi is the velocity field. Is working? Okay. Good. Is the velocity. Now there are two types of boundary conditions that you can apply. One is this psi is constant, say on the border, and this is the free part of the boundary where say the tumor can expand freely. But of course, during growth you can encounter obstacles, say a bone or another tissue, and so on. So there you need to apply, say, if the bone is rigid, it's a rigid wall, you have to apply that the velocity there is 0, which means that n dot v equals 0. Why I'm saying n dot v? Because it can slide on the wall, it cannot penetrate, but can slide on the wall. Okay, so this is the easiest, say, model for a tumor growth, or for anything that can grow in this setup. Okay, but of course, as I told you yesterday, you can repeat that also for other constituents, also for other cells, okay, and you can write down all sorts of mass balance equation for the things. For instance, let me mention something. One classical assumption that is also okay is to say that the extracellular matrix is rigid. You have seen from the first movie that I showed that this is not true, because the cells use the extracellular matrix to crawl, and then it deforms the extracellular matrix. But you know, as a first approximation, you can assume that that is rigid. If you are thinking of a culturing cell in a scaffold, well, that's even more true, because the scaffold is really rigid. Okay, so this means that Vm is equal to 0. So for the extracellular matrix, you can replace this general mass balance equation with just an equation with a time derivative and the growth term. Okay, some assumptions that are usually made on this set, on this framework, on this set of models is the following. The first one is the saturation assumption, like this one, okay? That is the population that you are interested in sum up to 1. And this means that if you add these two equations, the time derivative goes away, okay? Because two equations, many equations. Because you have the sum of the derivative of the sum of phi alpha, which is 1, which is 0, and so you are left with this thing. Okay, then I already commented about whether the mixture can be closed or open. Okay, closed means that the mass exchanges can only occur between the phases, and you don't have this. Okay, so if you have a tumor that is growing in a liquid environment, it is growing because it's sucking the liquid from the environment. This is what happens in a petri dish or whatever, where you don't have vessels in vitro experiments. Then there is another sorry, there is another assumption that is usually made, which is called in mixture theory the constrained mixture assumption. It tells you that all the velocity of the constituent of the cells is the same, okay? Which is this one. Okay, there is no index here. Which means that the two cell constituents in this case, in this one, they just move together. There is no relative motion between them. Okay, this is might be okay, for instance, for the cells, but if you have cell and extracellular matrix, this is not okay, because the cell will move with respect to the extracellular matrix. If there is the liquid, this is not true again. I mean, it's an assumption that is any assumption. I mean, is advantage and limitations in some sense. Okay, why this is also useful? Because of the same reason. I mean, if you look at this sum, if this velocity is independent of alpha, then you can take it out of the summation, and then you're left with the sum of phi alpha, which is 1, and then you are again in the same framework that you had before. The divergence of V is equal to 0. Okay? This V is the common velocity. 0 if the mixture is closed, or is something if it's not. And so you can use the potential flow assumption again. So in some sense, the constrained mixture hypothesis together with the potential flow assumption gives you an equation which is I wrote it before, that Laplacian of psi equal to something, which gives you the velocity field. And then a posteriori you find the evolution for the different constituent. Okay, this is an example of what you can get with these easy models. Okay, of course the model is sorry. Let me show the last one. Probably this one is okay. Yeah. Okay. This is by from the group headed by John Lovengrub, and you have this structure that is formed, this instability which resembles an instability which is called Saffman-Taylor instability. That's a part of the brain, and you can play with that. Okay, so I wanted just a bit to justify this disclosure, for instance, or to justify what is called Darcys Law. You have noticed that I didn't use the word Darcys Law because it's a bit confusing, at least for the pedagogical point of view. Probably at the end I have to give up. But because it's usually called Darcys Law closure, but Darcy has to do, as we will see, with the flow of the fluid and has other consequences. So, if you want to go to a step further without, say, making a closure which looks like, say, artificial, or if you want to justify that, you have to write down a momentum balance equation. Okay, so things becomes a bit more difficult to see, but let me try to explain all the terms in this. Okay, so this is the momentum, the density times the percentage of volume occupied by the cells, and the velocity, m times v. So you make the integral. So this is essentially the mass multiplied by the velocity, and you ask why this momentum should change in time. In that volume, that control volume that you are looking at, the microscope, the field, the microscopic, in the field of the microscope. Okay, so there is one thing that is related to, say, the flux, because if there are cells that enter with some velocity, you will compute an extra momentum, because before you were counting only the momentum inside the field, and then you have another car that is coming in with a certain velocity, so you will add another momentum. Then there is something that is related to the stress at the boundary, in the sense that you have a force coming to the boundary because outside there are other cells that are pushing, and so pushing means that you have an effect of the momentum of the cells inside. And these two terms are classical. These three terms are classical in continuum mechanics. But in this case, you have other terms, which are also this one is classic, because this is just the volume, the volumetric force, something like gravity, if you want. In biology, it's more than gravity. That can be, say, chemotaxis, and I will explain that later. But these two terms are very new. This, in some sense, it's a bit easier to understand, though it's often forgotten. This has to do with the fact that if you have a cell which is moving, and at some point, for instance, disappears because it dies, okay, you lose some momentum because it was moving with something that you don't see anymore, and so you're losing something, okay? Or if otherwise, you can do the reverse for a growing cell. And this is crucial because this is the interaction force of the cells with the other environment, with other part of the environment. Let's say the extracellular matrix, the liquid, and so on. The other cells, if you want. Okay. So that's the entire integral balance equation. And with usual tools, you can reduce that one to a local form. The usual means put the derivative inside, use Gauss theorem, the divergence theorem, and then localize the thing. And then you have an equation like this. This is very complicated in some sense. It's not too new with respect to the classical one. It only contains this new term, this interaction force. Now, luckily, in biological, and you can do that again for all the constituents that you want. Okay. Luckily, in biological, most biological phenomenon, especially in the growth phenomenon I'm interested in, or in motion, you can forget about inertia. Okay? Motions are very slow. And also the dimensions are very slow, dimensions of the cells. So the Reynolds number, for those who are familiar with fluid mechanics, is very small. So you can forget about this, okay? And usually you can also forget about the force. In the following I will not, but in one case, but it's okay. So at the end of the story, in a nutshell, mixture theory writes down as a set of mass balance equations and a set of, I would say, force balance equations, in which the stress is balanced by the interaction force. So the difference between these two terms is the following. This is a term that takes into account of the interaction within the constituents. That is, I'm in a crowded bus, okay, and so I'm moving because all the people are pushing me because they want to go out. That's in here, okay? This one has something to do with the interaction with other things. Something like, for instance, the extracellular matrix. Having said that, let me explain how classically from this momentum equation you obtain Darcys Law. Which was obtained, say, more than 1 century ago from experimental evidences, I would say. Okay? And from this general framework, that by the way was done 50 years later, again, you forget about the inertia. Let's for a moment forget about this body force. Okay? And you have these two terms. And the assumption that you can do on these two terms are that the stress is just proportional to the pressure times the identity matrix. T is the tensor, so Pi. Phi L takes into account of the volume ratio. And the velocity, the interaction force, is proportional to the velocity of the fluid. Of course, I mean, you expect that if you have a fluid that is moving to a sponge, to a porous material, then probably, if it is at rest, then there is no force. If it is moving, the first assumption that you can make is that it's proportional to the velocity, and of course, it is opposite to the velocity. It is a sort of drag, okay? So if you balance these two and you explicit VL, what you find is this one. So this is Darcys Law. Okay, Darcys Law in fluid dynamics tells you that the velocity is proportional to the gradient of pressure through a tensor which is called the permeability tensor, and here at the denominator you have mu, the viscosity of the fluid that is moving, because of course you can understand that if oil is moving in a porous material, it will be slower, and if it is, say, if it is water, it's moving faster. Or if you want, yeah, I forgot this term here. There should be a mu here. Of course, there is a mu here that I forgot. the drag force is proportional to the viscosity. Okay? But other assumption can be made. One that is also very interesting from the numerical point of view is the Brinkman equation, in which you just simply don't neglect inertia, and you assume that the stress tensor, this stress term is this one. Okay? It's just what is the difference between the two? The difference is that in one case the liquid is treated as an inviscid liquid, and in this case it's treated as a viscous liquid in the stress. Okay, and this is called the Brinkman equation. Okay, now let's look at more or less the same thing for an unsaturated bunch of cells. That is, I'm taking a model in which I have cells, and if you want, I have extracellular matrix that is fixed, is rigid, it's not moving, and I want to do the same thing. So I have this equation here. I kept for a moment this body force. So I have the stress of the cell, this interaction force, and this one. So, and in order to close the system, I do the simplest assumption that I can make. I have a bunch of cells, and I have to say, okay, what is the behavior of this bunch of cells when I compress them? What is the response to compression? And the easiest assumption that I can make is that it behaves like a gas. Of course, you can understand that this is very rough. The cells don't behave like a gas or like an inviscid liquid, a compressible liquid. But this assumption allows to write that this T is some scalar quantity times I. And as before, I'm assuming that this interaction force between the cell and the extracellular matrix, when they are pulling, they are moving in the extracellular matrix, is proportional to the velocity with the motility tensor. I can call a motility tensor, K now. As an example of body force, I might say that there is a force that is pulling the cell that is proportional to the gradient of some chemoattractant. This is usually in biological phenomena. You can have some effect of some, for instance, chemical field or other stuff that tends to pull the cell toward the maximum of this landscape of C. Okay, so again, if I play around with these three terms, in particular, if I express it with C, I have this. Okay, I have a way to find VC as a gradient of phi plus, if you want, if it is present, some chemotactic term, and I can substitute that into the mass balance equation as I did before, and I have an equation which is this one. Okay, so this is the basic equation for treating, say, ensemble cells as porous materials. Okay, it's a parabolic equation because this is a function of phi, nonlinear, degenerate because it has this phi c here. So the difference is that it has a boundary. If you start with a compact support, your solution will have always a compact support. And then this term, this advective term, the advective because there is this phi c here, describes the motion toward the maximum of this chemical field. Okay? And this is a generalization of a Keller-Seeger model that can be obtained under some assumption that, to tell you the truth, are very unphysical, I would say. However, this is the structure. Okay. So let's go back to a model like this. So if I have two now, if I have two cells with a saturated that are saturated, okay, then phi1 and phi2 sorry, and I assume that the velocity is the same. Then in this case the velocity is the same. I need only one equation of the momentum, and this might be this one. Okay, this equation here. Now, here there is a conceptually a big step that I want to clarify, and it's the following. In the previous model, you have an unsaturated case, and you can compress the fluid as you want. Here you have a saturated case. You remember that in this case you have something that divergence of V equals 0, so divergence of V equals something, and you cannot do that. When you have a constraint which is a geometric constraint on a material, for instance, take a fluid, an incompressible fluid, and you say that it's incompressible, no matter what pressure you put on your fluid, you won't change anything. It will not compress. It will respond to that with the reactive force, okay, with the reactive force. So this means that there is a Lagrange multiplier into the stress equation that is balancing this pressure that you are applying, because you are imposing this geometric constraint. So in some sense, you have something like this. You have a geometric constraint. Constraint. I don't remember. Geometric constraint, say volume fixed. This implies, say, for something like divergence of V, for instance, equal to 0. And this implies that the stress has a pressure term which is a Lagrange multiplier. Okay? And this is what happens in continuum mechanics. You have in fluid dynamics determinant of F equal to, or if you want, say, you take a volume, and the volume before and after the deformation is the same. Then this implies divergence of V equals 0, and this implies this one. Okay. And the same thing happens here. Here the constraint is this one. Sum of phi alpha equals 0, equal 1. Then there is something like divergence of what is called the composite velocity. And this tells you that there is a constraint in the stresses. Something like that. Okay? So that's why I don't like to call that closure over there Darcy's closure, but it's something that is related to this lambda. But this is essentially what you do. Okay? Then you use this V equal to some gradient of lambda or gradient of C if you want, and you put it back into the equation. Okay. Now, at this point, I have all the basic stuff, and I want to go back to the original problem. Okay? That is, this is just the equation for one single cell, the ensemble of cells in this spheroid, in an extracellular matrix, which is assumed rigid. Okay? It's assumed rigid. And I said it before, I'm assuming that this is proportional to the velocity through some permeability tensor, or motility tensor better. And so I have that Vc is equal to some motility times the divergence of T. Okay? I'm doing the same thing for this model. But now I want to use what I learned from the microscopic picture, from the microscopic models, and from the experiment that I showed you at the beginning. In particular, I know that the speed, this one, It's just not, it's just something that is proportional to something, but there's a threshold value. And this has some implication on the forces and on the permeability, on the interaction force and on the motility K. Okay? One has to say that if the area of the extracellular matrix is below a certain threshold value, this one, A0, then there is no motion. This plus is the positive part of the parenthesis, so it's 0 if the argument is negative. Okay, if AM is below A0, there is no motion because you are here. And then you start, say, for instance, as a straight line. So the velocity is proportional then to the difference between AM and A0. So this function here mimic this, say, discontinuous function. Of course, this might be not true in the sense that we have also learned that at some point this as a maximum is saturated. It saturates, I mean, when there is no extracellular matrix. Okay, so the pores are very wide. Okay, you cannot expect that the velocity goes to infinity, so it's limited. And probably, as you have seen, it's also decreasing. I mean, it has this bell shape. So there might be a denominator here with a power n, which is probably greater than 1, so that you have a biphasic behavior. So this is your closure. It's what we have learned from the experiments that tells you that the permeability is not just a constant, but has to do with some quantity that are measured, say, at the microscale. Okay? Not only that, but we have learned that A0 is not constant itself, in the sense that this critical value that decides whether the cell will pass or not depends on a lot of things. Depends on how big the nucleus is, then depends on the elasticity of the nucleus, the stiffness, or in the ability of the cell to exert traction forces, or again, I didn't show that, but of course, at the end I will show an application of that by the action of metalloproteinases. Because metalloproteinases is a protein that is produced by the cell in order to cut the fibers. So cutting the fibers means that you are, with a machete, you are going by enlarging, say, your path. Okay, enlarging this A0. So if there is, the cell is able to express this protein, this A0 will change with time. Okay, so there are a bunch of things that can influence this A0 there. So how this, is this function A0 related with what we have learned? For instance, is related with mechanical characteristics? Well, through this curve here. I mean, we mentioned this pore dimensionless, this ratio between the pore radius and the radius of the nucleus, which of course is just the same thing as the ratio of the area of the pore with respect to the area of the nucleus, the cross section of the nucleus, and this G has to be related now with the stress tensor. Okay. So we take the trace of the stress tensor. G is a scalar. Well, the trace divided by mu. Okay? So let's look at the simplest model, just one population of tumor cell, just tumor cells in the environment that are crawling. So the blue stuff that you see here is just an ensemble of cells that are growing in an environment in which you have a jump in the density of the extracellular matrix. Okay? And they grow, and when they find this region with a thicker zone, they are not able to pass. Okay? So let me mention that this was not possible with the model that were done before, because any time that you put a net in this kind of models, no matter what, if you don't take care of this, this is called physical constraint of motility, you have that the cell will pass through. Okay. Instead, by lowering, say, the density of the extracellular matrix to, say, 25%, yeah, 25%, then the cell will grow, and when they reach this denser area, they are able to pass. With some difficulty probably, but they are able to pass. Okay? This is a two-dimensional simulation. You have the area that is here with a higher density, 30%. Here there is, say, 20% again. And you have the bunch of cells that are is growing, the ensemble of cells. By the way, in this case, you have two populations. One is the normal population you don't see here, which that is filling all these regions, okay? And this is the tumor population that has a clonal advantage, and they continue to proliferate. But you can see here that when they reach this membrane, they don't go through, and they prefer to move in this direction. So it's like in a duct. I mean, in a duct, you have the cells that are constrained in the duct, and as you expect, if the tumor is growing along the duct. It's not passing through the duct, through the duct walls. But if you lower, again, the density of the extracellular matrix, if you want density, I'm saying density, but it's just you enlarge the pore area of the extracellular matrix, then the cells are able to pass. But more importantly for us is what happens when you have a stiffer nucleus and a softer nucleus. Okay? Here is the same as before. Now we soften the nucleus. This is a cross section in this direction, like here. Okay, more or less. The red are the tumor cells, and you find the blue cells are the normal cells. They are populating the inside and the outside, but they don't communicate. Okay, they cannot pass through this thick membrane because their nucleus is stiff. Now I'm adding a clone in which the nucleus is softer, and then when they populate and grow and they reach this area, this region here, then they populate the thick area, and they pass through and they go to the outside. Okay? So this population is able to do that. But as I told you, I mean, you have also to take into account of the stiffness of the membrane. You can add that to the picture because the stiffness influences the stiffness of the membrane, of the nuclear membrane, influences this H0, this critical value of the pores that allow passage or not. And here you can see that where the stiffness is very high, again, then the cell will grow and will stop at the region where there is a higher density of extracellular matrix, okay, regardless of, say, the nuclear membrane stiffness. Vice versa, if it is too soft, regardless of the nuclear membrane stiffness, the cell will go through, and in between you can have passing or not passing according to the nuclear stiffness. Okay, so these two ingredients, the stiffness of the nucleus and the stiffness of the membrane, play a role in passing through thicker region. And of course, you can play with what you like. I mean, making an environment which is very heterogeneous with the forbidden areas, thicker areas, and so on, and your virtual, say, ensemble of cell will find the path through this labyrinth. Okay, good. Well, good, but it's not really so good because in this model, what I did is to change, say, the pore area in very thick regions. Instead, these basal membranes are very thin regions. So numerically, it's not very convenient to use, say, something that is not even micrometer scale, it's even nanometer scale, and do a a mesh that is so refined. Okay, so you want to understand how to model things like this, in which you have the inside, then some thin membrane, and the cell can pass or not pass through this membrane according to the characteristics that I mentioned before, and the inside and the outside are separated by an interface which can be modeled as a surface. From the conceptual point of view, I think that you can understand that I have to replace this behavior, what happens in this membrane, by an interface condition, okay? That is telling what is communicating to the outside what's happening inside and vice versa. So this is the idea. So I'm starting always now with this simple model. Mass balance, and this is related to momentum balance, or if you want, to Darcys Law, but it's Darcys Law in which this pressure here is not the pressure of the liquid, it's the stress in the solid. That's the main difference, okay? It's the stress in the solid, there's the response of the solid to compression, to the cell to compression. And in general, before, I had a setup like that. There is an outside, an inside, and there is a region in which I have a thick membrane. Here, okay? So the problem, I hope you can see, is the following. I write the same equation as before in the three regions, inside, outside, and in the membrane. And of course you need some interface condition at this boundary, and at this boundary here, at the interface between say this, D1, D2, and D3, okay? What are the interface conditions? Well, of course, this can be proved, of course, mathematically. If you have a cell that is crawling and going from here to here, then the outgoing flux has to be incoming flux in the other domain. This is called the continuity of fluxes. Across interfaces, you have to impose continuity of fluxes, or if you want, whatever is in divergence here need to be continuous. Okay, this one. And you find it here. Of course, the normal component of that. Okay, this is the continuity of fluxes through this interface below, and the continuity of fluxes through the interface above. What is this second interface condition? This one is related to the continuity of forces, in the sense that if a cell here is pushing against a cell here, the cell there is feeling the push with the same force. I mean, you have transfer of forces, okay? And this is just this one. This is continuity of stresses. So these are the two, continuity of fluxes and continuity of stresses, that you can impose. There's something that should stay. Well, okay, never mind. Now, you can go to the limit, making this thickness of this membrane going to 0. And what you find out is that this problem becomes a problem in which the equation inside and the equation outside are the same, and that's what I wanted to say, okay, are replaced by these interface conditions. Okay. Now, what I wanted to say before is the following. Look here, you have a parabolic equation, inside, outside, and inside, okay? And this means that you need to impose, for a parabolic equation, you have to impose one condition here, one condition on the other side, one condition here, and one condition here. That's why you have 4 conditions. Okay, this is okay. If I'm squeezing this in 2, what I need is one condition on the top of the thin layer and one on the bottom, and these are the 2 conditions, 1 and 2. Okay, so this is enough to close the system. Okay, this is a strange equation in the sense that this part here is classical, in the sense that you recover, say, the continuity of fluxes. Of course, I mean, if you are squeezing this membrane, this thick layer to a membrane, to a thin membrane, of course, what is coming out of the membrane is going in to the other domain. Okay, this is again, but this is equal to some quantity that is a strange, as a strange parameter, pi, which is the derivative of pi is n pi prime, p prime. Okay, so it's a strange term, but it can be understood in, oh, well, okay, I can forget about that. You can generalize that to a lot of equations, a lot of, say, populations. But you can generalize that, you can understand that by looking at some special case. So this is the problem, the membrane problem. You have an equation inside and outside. You have a continuity of fluxes, what is inside the divergence, and then you have this extra condition. Okay? Now, to understand that, assume that this dependence of the pressure on the density is logarithmic. Okay? Of course, it doesn't make sense, in the sense that you have an infinite pressure when the density goes to 0. Negative infinite pressure when the density goes to 0, and it's concave. But let's do that. Why am I doing that? Because if I do the prime and p prime, this becomes capital P over n, and the two n cancel, and so this is just a constant, and so this jump is just what? The jump in n. Okay? This is n times p divided by n, so p prime, capital P prime is equal to p, so p is p capital P n, and so you have an equation like that. I forgot a p here. Okay, so this is something that someone might have encountered in his life. It's a Robin type equation, a Robin type boundary condition. It's called Kadem-Kasharsky equation, interface condition. This is used commonly for the diffusion of molecules through membranes. Here we don't have molecules, we have cells, but it's just the same. And of course, this is related to the fact that originally, in the diffusion equation for the molecules, you don't have this nonlinear term here. You don't have n, the gradient of P, you have just the, say, Laplacian of the concentration of the molecule. So the nonlinearity, say, goes away. And so for the same reason, this nonlinear term goes away and becomes just a constant, or a linear function. This is a constant and this is a linear in the concentration. Okay, so if you want, this strange interface condition is just a generalization of Kadem-Kasharsky interface condition to the nonlinear case. Okay, good. Let's see now how it behaves then. Okay, so this is a dumb simulation, 1D. It has no, say, biological meaning, to tell you the truth. It's just the steady state of something that is growing at a certain rate, reaching 1 with the logistic law. You know the logistic law now. Everywhere, then there is an interface, and it's dying here. It's absorbed here. And then if the thickness of the layer is not so thin, then you have this one. If you have a membrane problem, it's this one. And you can see that they are quite close, but if you want to quantify that, just measure, say, these quantities. Say the density at one side of the thick layer and on the other side, and the density on one side of the membrane and on the other side, and then compute the difference. And if you compute this difference, you have that the model, this model here, converge to that one linearly with the distance, with the thickness of the membrane. Okay, so if the membrane is very thin, you are allowed, actually it's much better to use the membrane model and that interface condition. So this is just a way to check that numerically, if you want, that the procedure the limit procedure was correct. This is probably a more sound biological, virtual biological experiment, because I have a region D1 and thick layer, okay, here, the two, and say the outside. At the beginning, there are tumor cells inside here, and normal cells outside. So the tumor cell will grow more, okay, and they will invade the outside. So this interface here, this vertical line, is the boundary between the tumor and say the growing stuff and the stuff that is not growing. And then you have this traveling wave problem, if you want, because there is a traveling wave going on. And you can compare, say, the solution here. You have the tumor, the jump, decrease, then it decreases again, and then you have the healthy tissue, and with the membrane problem. Here there is just the membrane, and you don't have this decrease, you have just a jump, but they look the same. They're very, very close. So this is again the behavior of what happens on one side and on the other side of the thick layer, and you can see that they are pretty equal. Apart from here, which is the time that is taken from the cell to go across the thick layer. Okay, so this is what we wanted to have actually, to have a model in which you can play with, you can introduce this membrane stuff in order to have compartmentalization of cells in compartments, within basal membranes. Okay, so in this thing, in this simulation here, you have say the growing tumors here in a duct, but there is a hole here. Okay, and so they will. A hole doesn't mean a real hole. It's just a region in which the pore size is a bit bigger. Okay, so here is forbidden. I mean, they could pass, but the pore size is very small. It's limiting, statically limiting, and here they are not. The pore size is so big that some cell can escape. And they can invade the outside, and you have the normal tissue in blue, compresses. This white stuff is the border between the, say, the tumor that is growing and healthy tissue that is compressing, is compressed by the tumor cells that is growing and is going outside. Okay, and I think that, and you can see a simulation here. Okay, at the beginning you see this flash because at the beginning here everything is blue, so the cell grow first into the duct and then they escape with a longer time, on a longer time scale. But then you can do what actually was asked a few years ago, I would say. It took a long time to reach here. Probably it's 3 years ago, probably even more than that. The original question was to describe, say, the behavior of pancreatic tumor and of these ovary tumors in this way. And we had to add metalloproteinases, because metalloproteinases is the thing that I mentioned before, is the in charge of opening the nets and allow the passage. How do you do that? Okay, for instance. Well, you can assume that there is a target pore cross-section, which is restored continuously by, say, fibroblasts, for instance. So if there is no metalloproteinases, the CMMP is the density, the concentration of metalloproteinases. So if there is nothing, then this equation will go to the stationary value A equal to A1. So it's continuously restored. Okay, but if there is metalloproteinases, this has the action of increasing the area. Okay, and we are taking that proportional. I mean, we don't have data on that. So the easiest thing to say is that just, for a constant value of C, then the area increases proportionally with the concentration. Okay, and we already know that the motility, now in the model I call it mu, I'm sorry for changing the notation. The motility has this threshold, A0, okay, and above the threshold behaves, say, at the beginning linearly, and then saturate. You can put an N here, it doesn't change much. Okay, and you can play with your favorite tissue. So this is an ovary tumor. At the beginning, the cells are here. They degrade this membrane here, and they start to go into the mesothelial liquid. Okay, they move, say randomly, but diffusing and compressing the things that are here eventually. And they are reaching this membrane here, and gradually they are degrading the membrane, so more and more cells will be able to pass and pass through the mesothelial lining here, that is this one, okay, and degrade the extracellular matrix below and invade also the layer below, okay. Unfortunately, the movie that I will show is a bit fast, but probably I can use, let's see, I can use this one if it's working. Should be this, because this one loops, okay. And this is the evolution. You can, for instance, appreciate the fact that this is becoming, say, more and more compressed, more dense, and it takes some time to pass through here. I mean, there is a slowdown because it's needed by the tumor cells to degrade and to allow to passing. So the first thing to do is that this ensemble of cells will just sit on the membrane, they will degrade and they will pass. Okay. Okay, so, yeah. Okay. But if you want to look at the same phenomenon, again, from a more, say, microscope cellular, at the cellular scale, then again, probably this one is not so good, because this is just tells you what happens to a lot of cells, okay? So if you want to look at the behavior of just a single cell on this mesothelial lining and passing through, then again, you have to use an individual-based model. And we use our preferred one, okay? So the mechanism is the following, if you want to look closely at it. You have the cell, the single metastasis, touches the endothelial line and the mesothelial lining, this lining of cells that are covered by what is called the peritoneal lining, a lining, say a small layer of extracellular matrix around the cell. Okay, so the first thing that they have to do is to degrade this this thin layer with metalloproteinases. then the cells are left without any protections, but they are still attached. They, through cadherin junction, they are very stiff. So the question is, how does the cell convince the endothelial cells to, say, separate and go through? Okay, and they do that through a protein that is called CD168. Okay, so that this loosen up the junction between the cadherin, and loosening up this junction, the cell is able to penetrate and go below. Why we were interested in that setup? Because let's see if I have experiment. Okay, I will show you that later. Because they are doing experiment in vitro on that. Okay, so they put just single cell or a bunch of cells, not too many, on this artificial mesothelial lining, and they are testing that in vitro by saying what happens if I interfere with the cell in this way, in this other way, and so on. Okay, so this is the biological picture. This is our, say, informatical picture, if you want, modeling picture. Okay, and this is the result of the simulation. We have the cell that is going down, is adhering to the extracellular matrix. It's degrading the extracellular matrix here. Is interfering with the cells, so that the junction means, if you remember the cellular post model, it has a term that is in charge of, say, an energy which takes into account of the interface energy, how the cell wants to stay together. So interfering with the protein means that you are injecting some molecules so that this term there decreases. Okay? So decreasing that term, the cell can penetrate like this and go through, and then the lining closes up. Okay? And so this is the movie related to what I showed. Okay. And this, in some sense, is the comparison. This is the experiments that they do. The green stuff, of course, is the bad guy. Okay? Sits over the lining, then it penetrates down and goes below. And the question now is, what happens? This is the say, in some sense, what as modeler, as mathematician, we can give to the biologist. I mean, try to test what is, say, most convenient to do as a first experiment. I mean, to classify the experiments according to what is more promising. Okay? So what happens if you change the parameter related with metalloproteinases? If you inhibit metalloproteinases, what do you expect? Of course, if you inhibit metalloproteinases, then this layer here will not be degraded, or it takes a long time to be degraded. And so it stops the process from the beginning, okay? So the cell cannot interfere with this CD168, it cannot penetrate. So probably this one is the most effective thing to do. If you, say, interfere with the interaction between mesenchymal cell, again, in this case, the cell is able to loosen up the junction, and this is not. So it's able to degrade the ACM on the top of it, but it's not able to pass through because this junction is too strong. Or the same thing happens if you interfere, of course, with the production of this molecule 168. Will stay there. If you increase the adhesivity between the cell and the mesothelial lining, what happens is that the cell will stay there because it likes the mesothelial lining, and in some cases it replaces the mesothelial lining. So it goes here, and you will find a blue cell instead of the gray cell. Okay. Will not invade the layer below because it prefers to stay there. So this is in some sense a dysplastic behavior. Okay. It's displacing the normal tissue in, by tumor cells. Okay, and you can do the same thing for say a bunch of cells. Okay, these are say the experiments. This is the top view of a bunch means say 20 cells, no more than that. And you have that passing through, and actually in this case is the destruction of the mesothelial lining because there are too many cells. This is the side view. Okay, you don't see here the cells any longer. And this is the bottom view. You have one single cell that is peeking out, and then the layer is destroyed. So the difference between, say, one single metastasis and, say, a collective motion of metastasis is that the second one is more destructive. Okay? And with the same model, you can do that, and more or less you get the same. Okay? You have the bunch, there are 10 cells here, whatever. Yeah, 10 cells. They eliminate the extracellular matrix, then they start to dig into the hole, and they are replacing the layer below. Okay, and this is the comparison with the experiments again. Okay, I could stop here, but as this is a summer school and you will be all involved in projects, and though I will not be here, I will just mention some topics that, according to me, are well, say, deserve to be studied in the future because nothing is done in this subject. So if you want to be interested in that, there is empty room, full of space to publish whatever you want, even bullshit. They're okay. Then someone has to say that it's wrong. I think it's okay. It has to do with a new topic which is called mechanotransduction, mechanosensing. If you want to have a definition, it's this one. The mechanosensing is how the cell feel the mechanical forces by the environment. And it's coherent with the general topic of my talk, because I was thinking about, say, what is the interaction of the cell with the extracellular matrix. Okay. Mechanotransduction is in some sense the reverse, in the sense that the cell feels the environment mechanically, and it responds to the mechanical environment, by expressing something. Okay, this is well known, but nowadays the interest of biologists is enlarging. Before, everything, all the biology was biochemistry, I would say. Here is at the interface between biochemistry and mechanics. For instance, if you have a bunch of cells like that in an extracellular matrix, and you apply a force, okay, then this force interferes with some transmembrane molecule or channels, for instance, so that the ion channel will open up, and so more calcium you have inside the cell, and this will change the behavior of the cell. Okay, or other things. There is a protein, for instance, called tallin here that is very nice, because under stress, what happens is, let's see, you will see it here, tallin. So I have the extracellular matrix binds to the integrin binds to the tallin, okay? And when it binds to the tallin and is pulled, wait. Okay, and when it's stretched, it opens up adhesion sites where different molecules can attach. Say, can phosphorylate, for instance. In this case, vinculin can attach. And this will change the property of material. So this is a sensor, it's a mechanosensor. Okay? I mean, what I'm saying is not new. Everyone knows that in order to heal a bone, you put in traction the bone. Right? If you have an astronaut, then because of the lack of gravity, his tissue will just die out, especially the bones. So the interaction between stresses and the behavior of cells is well known since, I would say, centuries. Probably not centuries, but decades at least. Now the interest is in understanding these mechanisms here. And why is that? For instance, going to tumors, it's known that according to the environment, cells behave differently. Okay, so if you put a bunch of cells, like the cell of a duct breast, okay, a breast duct here, in normal conditions they just behave normally, just the same cells. But if you put them in stiffer gels, they become hyperplastic, and their invasion characteristic changes. So there is a new paradigm that is now open to exploration, is that probably it's better to cure the tumor by, say, normalizing the tissue around it. One thing that was, for instance, one question that was posed by a guy that was called Rakesh Jain, compared to anti-angiogenic therapy, that, by the way, are found to be, say, in some sense, not so good, okay, for several reasons, is that one should restore the vasculature, the proper vasculature in the tumor, rather than destroying it. The same thing can occur for the extracellular matrix. The tendency of the tumor is to, say, stiffen up. And also the tendency of the environment, I mean, on the environmental cell of the other cells, is to, say, stiffen up the region around the tumor. And everybody knows that tumors are stiffer tissues than normal tissue. This is the way a woman, say, feels a nodule in the breast. Okay? You feel something in your lung because the tumor is calcified, the environment has a lot of extracellular matrix, so the environment is stiffer than normal. And this will induce, say, metastasization. Okay, so this is the experiment by Weaver's group. Okay, this is the normal, say, acini of the breast. Then you increase the elastic modulo of the gel in which you grow in vitro this ensemble of cells, and then they change phenotype, actually. They change phenotype. They are from being epithelial, you go mesenchymal. They just don't form anything, and they start to crawl and to move around. Of course, you have to change a lot. But there is probably, oh, okay. And this is a list of all other diseases. I don't want to stick with tumors. I mean, in some sense, I'm sick and bored of tumors. I've been doing that for 20 years. But there are a lot of diseases that have to do with the incorrect perception of the stresses of something that happens to the extracellular matrix. Okay? To name a few, say atrial fibrillation, abnormal conversion of mechanical stress into gradient of electrical activity, intimal hyperplasia, stretch activated signaling cascade due to stents and grafts. This has to do with the cardiovascular diseases. Or say dementia also seems to be related with an overpressure of the liquid inside the brain, and so on. You see the list is incredible. All the fibrosis have to do with the excessive production of extracellular matrix, and so on. Okay? But there is another thing that probably is more interesting than that. Nowadays, the key topic is to build a tissue in vitro. Okay? In order to do that, you have to seed stem cells in vitro. Where? In the nice environment. In the environment they would find in the body. If you put a stem cell which is not differentiated, can be anything, is pluripotent, okay? It can become a neuron if the elasticity of the matrix is soft, if the matrix is soft, or can become a bone if it is too stiff, or can become a muscle, or whatever. The same cell. So you put a cell, according to the environment, it changes the fate of the cell. This is called mechanoreciprocity. Okay? Let me mention. So this is, for instance, one reason why, in principle, it would be very nice to have, say, the cure of, say, brain damages from Alzheimer, from even from car crash and so on. But the problem is that when you have a damage on the brain, you have a scar. And the scar is, of course, stiffer than the tissue. The tissue, the brain tissue is one of the softest one. It's the softest one. Probably like Nutella. So what happens is that if you seed there a stem cell, it will not become a neuron. It will become a muscle. So it's useless. Okay? Instead, for the heart, you have now they are constructing heart patches in right environments, because they can be done, so that you can heal, say, damaged part of the heart. Okay. So this is the trend in biomedical engineering, I would say, but I would say that also that my feeling is that they are moving like, say, before Galileo, okay, in an empiric way. Let's do this test, let's try this, let's try that. There is no model on that. There is no, say, highway to. There is an highway actually to be built in order to speed up this thing. So I encourage everyone to look into this problem. Okay, and thank you for the attention.